Hello and welcome back. So today we'll be learning about nucleosomes. In the last class, we learned about different degrees of compaction of DNA. In that lecture, we learned that the first level of folding of DNA is known as chromatin. It is also known as beads on string structure, which is 11 nanometer fiber, and it looks something like this. Okay. So this beads and string structure, you can see something is repeating itself over the chromatin, and this repeating structure, this repeating structure is known as nucleosome. This is the structural unit of the chromatin. So what is nucleosome? Nucleosome is actually DNA protein. That protein is histone. So it is DNA histone complex which repeats itself over the chromatin. So it forms the structural unit of chromatin. So it is the basic unit of chromatin. Now, what is tissue made up of? Tissue is made up of a lot of cells, multiple types of cells. So what we can say that cell is the basic structural unit of a tissue. In that way, nucleosomes are the structural unit of chromatin. So how researchers got to know that nucleosomes are the basic structure? They are the structural unit. So for learning that, we have to take a look at an enzyme which is known as micrococal nuclease. So micrococal nuclease is an enzyme which is which has both exo and endonuclease activity. So what are those? Okay, so nucleases are the enzymes which can cut DNA or RNA in a certain manner. So it actually cuts DNA and RNA at the phosphodiester bond of the backbone. Okay, so what both exonuclease and both exo and endonuclease, they cut the DNA or RNA at the phosphodiester bond. So why are those two types? So exonucleases. So assume that this is a DNA. So they cut DNA from a certain end. Okay, either they can cut DNA from this end or this end at their sites. Assume that these are the sites 1, 2, 3, 4, 5. These are the cut sites, means the sites at which DNA can cut. Okay, so these are the cut sites of an exonuclease. So DNA can be cut only in the sequence 1, 2, 3, 4, 5, or it can cleave the DNA at 5, 4, 3, 2, 1, either from this end or from this end. So it can have its activity from 5 prime to 3 prime direction or from 3 prime to 5 prime direction. While endonucleases, they can cut DNA at any site. I mean, they can cut DNA randomly. They can cut at second, then they can cut at 5, they can cut at 4, 5, and 3. So they can cut randomly at any sites. They can be sequence specific or non-sequence specific. Okay, so those are the endonucleases and exonucleases. Endonucleases and exonucleases. So both of these activities, MNase has both of these activities, endonuclease activity and exonuclease activity. So for this is the micrococal nuclease. Okay, this is the micrococal nuclease. This is also known as MNase. So this is MNase and this is the other MNase. Okay, so these are the MNases. So what happens is MNase must cover the DNA. It must cover the whole DNA for it to cut it. Okay, so it should be covering the whole DNA, and only then it can cut the DNA. Okay, so what happens is at the nucleosome, so this is the nucleosome. It is a DNA histone complex, right? So this is histone protein around which the DNA is wrapped. This is DNA, and this is the histone protein. Okay, So what happens is that the DNA at the nucleosome core, so this is nucleosome core, it's the nucleosome core, the DNA is protected. Okay. The outer surface of DNA is exposed, but the inner surface of DNA is protected by the nucleosome core, and that's why at this point MNS can't cut. Okay, so let's see how this helped scientists to decipher, to discover that nucleosome might be the structural unit. And then it is a now well established fact that nucleosome is the structural unit. So what happened was researchers separated out chromatin. So this is chromatin. Okay, so how will it look? It will look like this. Okay, so this is chromatin. And they treated chromatin with MNase. They treated chromatin with MNase, micrococcal nuclease. Okay, so what will happen now? So upon treatment of MNase, we'll get a lot of DNA fragments of different size. So where can cut the MNase? MNase can cut here, it can cut here, it can cut here, again it can cut here, it can cut here. Okay, again it can cut here, here. Okay, so what will be the result? We will get either mononucleosomes, dinucleosomes, trinucleosomes. A lot of such combinations are possible. Okay, or tetranucleosomes. So these are the possibilities. So what happened was when those DNA fragments were run on gel electrophoresis. So this is a technique by which we separate the DNA fragments. So assume this is a slab of gel. This is a gel slab, okay, and this is a well. In this well, we loaded the DNA fragments, and now DNA fragments are separated. So the separated DNA fragments were of certain sizes. So now this is 200 base pair, this is 400 base pair, this is 600, and so on. The base pairs. Okay. So the thing was that those fragments are repetitive in length. The 200, 400, 600, they are in multiples. They are in multiples of certain length. Here they are multiples of 200 base pairs. Okay. So this states that from this researchers concluded that chromatin is made up of something which is repeating itself over the length, over the whole chromatin, okay, over the whole length. Okay, so this something which is repeating itself over is nucleosome. I hope you get it. All right. So this is how nucleosomes were discovered. Now let's take a look at what are nucleosomes made up of. Okay. So nucleosomes, they consist of nucleosomal core. This core is made up of histone proteins. Okay. I already told you. This core is made up of histone proteins around which DNA is wrapped. So 146 base pairs of DNA is wrapped around nucleosomal core. And this DNA makes 1.75 tons around the 1.75 ton around the histone core. So this is the nucleosome core made up of histones. Okay, so this is made up of histones, and around this DNA is wrapped. So this is DNA. So it will wrap itself up for 1.75 times. Means it won't complete the 2 tons. So let's assume that this is the starting point. From here, the DNA came and it wrapped around itself. This is 1 ton, and this is 1.75 ton. Okay, something like this. So it wraps around the histone core in 1.75 ton, and it is made of 146 base pairs. So the nucleosome core is made up of histones, 4 types of histones: H3, H4, H2A, and H2B. These four types of histones are present in twice. They are present two times in the histone octamer. See, octa means eight. So something which is made up of eight subunits. So it is made up of eight histones. H3, H4, H2A, and H4. So this is the H3. Okay. This will be the H4. So this is again H3. This is H4. This is H2A, H2B, and behind it is again H2A and H2B. So assume it's like this: H3, H4, H3, H4. Okay. This is H2A. This is H2B. Here H2A again and H2B. This makes the histone octamer. So this is a 3D arrangement. Okay. So around it, this DNA is wrapped. You can see the DNA is wrapped around the histone octamer. So this is the nucleosome. So what happens that this packs the this packs the DNA. But for efficient packing, there is one more histone which is known as H1 histone. This is also known as linker histone because it is linking the one nucleosome to the next nucleosome. Okay, that's why it is known as linker histone. This is H1 histone. H1 histone plus nucleosome is known as chromatosome. What is this called? This is called as chromatosome. Okay. So what will be the final structure of chromatin? Okay. So this is DNA. This is the histone core. Around it, DNA is wrapping up itself. Okay. This is next histone core. DNA is wrapping up itself. Next histone core, and so on. Okay. So when DNA wraps itself around the histone core, here comes the linker DNA, H1. Oh, sorry, here comes the linker histone, H1. And the DNA between the two nucleosomes is known as linker DNA. As the name suggests, it is linking the two adjacent nucleosomes. Okay, this is linker DNA. And this H1 plus nucleosome is known as chromatosome. So this is how chromatin is looks like. This is how chromatin is made up of chromatosomes. Okay, so I hope this was clear. So now let's take a look at what are histones made up of. Okay. Oh yeah, one more thing. Um, so 146 base pairs of DNA wraps around itself on the core, on the histone core, right? So when linker DNA comes, H1 histone comes, not linker DNA, H1 linker histone comes, it wraps around at least 20 base pairs more. Okay, wraps around at least 20 base pairs. It can wrap 0 to 80 base pairs. Oh, sorry, 20 to 80 base pairs. 20 to 80 base pairs around itself. And thus, we can have structure known as chromatosome. Okay, so now let's take a look at histones. What are histones made up of? Okay, so histones are proteins. Okay, so proteins have different domains. So they are made up of central domain, which is known as histone fold. This is a central domain, histone fold domain, and they will have a C-terminal domain, the C-terminal extension, and an N-terminal domain, which is known as N-terminal histone tails. Okay, so what happens is, okay, let's have a look at this diagram. So when this is the histone, okay. So all of the histones have their N-terminal tails protruding out of the nucleosome. Okay, and that's why they are called tails because they are protruding out. They are nothing but stretch of amino acid which portrayed out of the DNA. Okay, these are very important because all of the post-translational modifications happens on histone tails. Okay, uh, one happens in histone core too. We'll discuss in upcoming videos. So N-terminal tails portrayed out of the nucleosome. Okay, so histones are made up of three domains: centrally located histone fold domain, C-terminal domain, and N-terminal histone tails, C-terminal extension. Okay, so these are how histones made up of. Now let's take a look at histone fold domain. Okay, so this is histone fold schematic. Okay, I made this for you guys to understand. So histone fold is made up of three helix. It made up of three helix. Okay, centrally located, and the biggest one is helix 2. Let's change the color. Yellow? How about yellow? No, let's take orange. Okay, so the centrally located and the biggest one, biggest helix is helix 2. And then at the N terminal, helix 1, and at C terminal, helix 2. Okay, and they are connected by strands. Strands are nothing but stretch of amino acids which connect those adjacent helix. So this is how histone fold looks like. So histone folds are very important because they are the ones around which DNA is gonna wrap itself up. Okay. So what happens is, so assume that this is the histone fold domain. Okay. This is a histone fold domain of histone 3. Assume this is histone fold domain of histone 3. And then this is the N terminal. Okay, sorry. This is the N terminal, this is C terminal. Okay, and this is other histone fold domain of another histone. Okay, and this is the N terminal of histone, and this is the C terminal of histone. So what happens is there are the N terminal domain of one histone fold and the C terminal domain of other histone fold, they interact with each other. The N terminal of histone fold and C terminal of other histone fold domain, they interact with each other, as well as this parts interact with each other. Oh, I'm sorry, I'm fumbling. So these three parts, the N terminal and C terminal, this is the second part, and these three parts interact with each other and forms docking surfaces for DNA. So we have three docking surfaces of DNA: one, two, and three. So around which DNA is wrapped. So have a look at it. You have N and C terminal interacting with each other. Okay, those two those strands interacting with each other. So you have one, two, three docking surfaces over which, assume this is DNA. So this the inner part of the DNA supercoil. This is the outer part, and this is the inner part. So inner part of DNA supercoil, inner part of DNA supercoil wraps itself around the three docking surfaces which are formed by histone fold domain domain of core histones. Okay, this is a very important statement. Take a note of it, that inner part of DNA supercoil wraps itself around the histone fold domain of the core histones. So that's it for today. In the next video lecture, we'll learn about chromatin remodelers. Till then, see you. Uh, okay, so I forgot to add one topic. Let me explain you how the histone core, or say, no, not histone core, let's say how nucleosome assembles. What is the order of nucleosome assembly? Okay, this is a very important topic. Let's discuss in this video itself. Okay, so what happens is first H3 and H4 forms a dimer. Okay, this is a very important topic. Please take a note of it. Please pay attention. The order is very important. First H3 and H4, those histones form a dimer. Okay, then what happens is other H3 and H4 forms dimer. Okay, so those are the H3H4 dimer, and again next H3H4 dimer. So what happens that the first H3H4 dimer comes on the DNA, sits over the DNA. So this is the DNA, this is the H3H4 dimer. Okay, then what happens is other H3H4 dimer comes, H3H4 dimer comes, which it comes and binds to the H3H4 dimer, which was already sitting on the DNA, and thus it forms thus so so so, so the H3H4 dimer which comes newly and the H3H4 dimer which was sitting on DNA, both of this come together and they form H3H4 tetramer. H3H4 tetramer. Okay, so after that, third step is H2A and H2B forms a dimer and it comes and attaches to this tetramer. Again, another H2A, H2B dimer comes and it interacts with this uh, this what do we call this? This is not called hexamer. This is I don't remember. I'll put the name in the description box below. So another H2A, H2B dimer comes and attaches with this complex, which is already formed complex. So So first H3, H4 dimer forms, then H3, H4 tetramer forms. Okay, then it comes H2A, H2B. Okay, and then comes another H2A, H2B. So this is I've drawn this in 3D. Okay, so H2A, H2B. This is again H2A, and this is H2B. So this is how a octamer is formed. Okay, that's why octamer, histone octamer, or histone core, or say nucleosome core. Let's not call it histone core, it's a misnomer, I guess. So, the nucleosome core, it is a tripartite system. Tripartite means made up of three parts. What three parts? H3, H4 tetramer plus H2A, H2B dimer and plus H2A, H2B dimer. Okay, take a note that only H3, H4 forms tetramer, not H2A, H2B. So it's a tripartite system. It has three parts, not two parts. No, logically it should form two parts because it can form H3, H4 tetramer. No, ma'am, we will say this is easy, but in bio, things are not that easy. So H2A, H2B tetramer, but it is not like this. It is not like this. Okay, so H3, H4 tetramer forms, then comes H2A, H2B dimer, and then comes another H2A, H2B dimer. So we have one, two, three parts, and that's why octamer is a tripartite system. This is very important thing that it is a tripartite system. Don't forget this. And yeah, the order of the assembly is very important. So H3, H4 dimer, then H3, H4 dimer comes and it forms H3, H4 tetramer, and then again H2A, H2B dimer comes and the other H2A, H2B dimer. So this is the This is the order, and it is a very important thing to remember. So, this whole process is assisted by histone chaperones. The chaperones are the protein complexes, which helps in the protein folding, right? So histone chaperones, the first discovered was nucleoplasmin. Okay, I'll discuss about this histone chaperones and chromatin remodelers in details in the next video. But now just take a note that these are the assembly of the nucleosome, nucleosomal assembly. It is assisted by histone chaperones as well as chromatin remodelers. In the next video, I'll show you a paper. I'll discuss a research paper which was published in 2005, okay, in the Nature. So that is a very interesting research paper which tells us how chromatin remodelers are also important in nucleosome assembly, and how I'll again explain an experiment which clarified the idea that histone chaperones are also involved in nucleosomal assembly. So till then, see you. Bye bye.